place in the sex organs, the ovaries and the testes in the animals, and the anthers and ovules in the plants. Note that the cell division in the nucleus is called the karyokinesis, and the cell division in the cytoplasm is called the cytokinesis. Now, in meiosis, we have 5 phases: interphase, prophase, metaphase, anaphase, and telophase. Interphase is where DNA replication takes place. And then prophase is when the chromosomes cross over. And then metaphase is when the bivalents stand at the equator. And then anaphase is when the spindle fibers shorten and they are pulled to the opposite poles. And then the telophase is when the cell division takes place. Thank you.
Greetings, everyone. Can we have IQ to come present the process of crossing over? You want to use the body? Yeah, you can. You don't want this. I just want to, like, erase. What's this?
Oh, no, it's fine.
A mic, so that you can be audible, like, yeah.
I can erase what's on the board. Using this. I won't use the mic, it's fine.
Good morning, everyone. My name is Vandina, and I'll be talking about the process of crossing over from IQ. Okay, can I have your attention? So crossing over happens in process 1 of Meiosis 1. In crossing over, there's 2 chromosomes that lie next to each other. One of maternal origin and one of paternal origin. So if the homologous pair, in the homologous pair, there's one, like this one, this could be of the maternal and this is the paternal. So the 2 chromosomes form a homologous pair, also known as a bivalent. So the actual point of crossing over is called the chiasma. And crossing over is the exchange of genetic material between chromates of homologous chromosomes. So after crossing over, each chromosome now has genetic material of the other chromosome. So each chromosome is made out of one original chromatid and one chromatid with the genetic material of the other chromatid from the homologous partner. So crossing over is important because it results in an exchange of genetic material. And therefore we can say that crossing over is responsible for genetic variation. Thank you.
Can we have Royal College to come present variation and causes? Variation by...
Okay, non-disjunction and its consequences. Royal Kings, Florida.
Non-disjunctions and... What?
Non-disjunction
And its consequences by Royal Kings, Florida.
Can I have a marker, sir? Must I find it? That's fine. Good morning, everyone. I'm Chandler from Royal King School, Florida. I will be doing a typical exam question that they ask in meiosis. It's a part of meiosis, non-disjunction and its consequences. So non-disjunction's consequence is basically Down syndrome. That's what we know. Because of the chromosomes, we know there's 23 normal, a normal functioning human being will have 23 chromosomes, not 24. So non-disjunction causes 24 chromosomes to be in the cell, basically in the normal person. And then let me just write this down for you and then explain it. So a basic question would be describe how non-disjunction may lead to Down. So basically, non-disjunction is the failure of this cord. It's causing me problems now. It's the failure of the strands to separate. What's this? It takes place in Anaphase 1 and 2. Non-disjunction during Anaphase 1 and 2 in humans results in an abnormal gamete with an extra chromosome being formed. The fusion between abnormal gametes, 24 chromosome, and a normal 23 chromosome results in 47 chromosomes instead of 46, leading to Down syndrome, basically. When spindle fibers fail to separate during meiosis, members of one pair of homologous chromosomes fail to become separated. During Anaphase 1 and may happen in Anaphase 2, two gametes receive an extra copy of affected chromosomes. So that's basically all you need to know about that question. Should I write it down still or did you all get it? Okay. Are you all gonna take pictures? Junction during Anaphase 1 and 2 in humans Abnormal. Yo, fuck, man. Being formed. The fusion. So yeah, so 24 chromosomes plus the 23 chromosomes, which will result in 47 chromosomes. The 24 chromosomes are an abnormal gamete, plus the 23, which is a normal functioning chromosome, equals 47. And then that will lead to a person who has Down syndrome, which is a disease or a dysfunction, learning disability. Basically, these people will be slower than normal human beings, their mind will not be able to process information as fast as a normal human being's person would be. Their cognitive abilities are slower than ours, basically. And that's all there is to know about this topic, basically. It was a very...
The lid.
I honestly don't know where it is.
I think. We are now moving. Oh, do you have questions? Contributions? Yeah. Teachers? Anything you want to contribute?
Good morning once again. Hope you're not going to get tired of my face. I want to begin from where this student ended, and I want to begin particularly here. Down syndrome is caused by an extra chromosome, which makes it 47. However, you must be clear on which chromosome pair. Okay? We must be very, very clear on which chromosome pair, because you also have a disease that's caused by an extra chromosome on pair 7. That's not Down syndrome. You'll have 47, but it's not what? Down syndrome. So Down syndrome only occurs when the extra pair has one other extra on chromosome pair 21. We must be clear on that. Okay? That's why I wanted to begin from. And then we go to the process of meiosis. We looked at meiosis 1 and 2. We are supposed to be able to define what meiosis is. Okay? Meiosis basically is known as a reduction process. Why do we call it a reduction process? And why does it occur in gametes? We know that gametes we have both from your father and the mom. Therefore, when the two fuse, we expect a normal human being to have 46 chromosomes. Okay? Therefore, it means if also the sperm cell and the ovum has 46, what will be the total? 96. Meaning you will be a normal human being, and we will call that as polyploid, meaning you have too much. So that's why in the gametes, they must be reducing the chromosomes from 46 to 23. Okay? That's why meiosis occurs. That's the main reason. That's why meiosis occurs. So that we don't produce a abnormal human being. Okay? So, what happens? Meiosis 1 is basically where the basic reduction occurs. Because you've already formed a zygote. This zygote has the 46. Okay? Meaning this cell has 46. You're supposed to reduce it to 23. What is the main difference between the two? The processes are all the same. But the process is that in meiosis 1, it occurs after a pair of chromosomes has replicated. Okay? Meaning you will have 4 strands. Okay? That's where you're supposed to realize the difference. 4 strands. But in meiosis 2, it's only 2 strands, 1 chromosome. We must be knowing that. Because when we just say chromosomes pair, they separate, we must be able to know where and how many are there. So in meiosis 1, you have a pair which has replicated to form 4 chromatids. So those 4 have to be separated into 2 on both sides of the cell, and then later, that's when in the last stage of meiosis, which is meiosis 2, the 2 chromatids separate into individual cells. Are we clear? I think that's all that I wanted to add. If there's another teacher or another colleague, I think we can. Thank you very much.
Thank you very much, sir. Please, can we invite the learner from RKS that wants to contribute to this? A round of applause for him, please.
What was this? Oh. Ma'am, I can't use this, please. Ma'am. No, wait down. I'm from RKS. I won't really say my name, ne? But no. I won't say my name, but I'm gonna do what I came here to do. I'm here to present meiosis 2, like the whole of meiosis 2. So I'm gonna start with. Someone, I think earlier, started with the phases, which is prophase, metaphase, anaphase, and telophase, right? Yeah. So, meiosis 2, did you guys finish meiosis 1? Like it's done, eh? It's class. Sure. So, yeah, meiosis 2, you basically begin with 2 cells, 2 individual cells with chromosomes inside. Yeah, this is prophase 2 of meiosis 2. This is where this is the centriole. Yeah. So in this process, this is where spindle fibers form. This is the formation of spindle fibers, and then you move on to metaphase, which is in the middle. So the centrioles are in the middle. Metaphase 2, where the spindle fibers have formed, and you have this is the beginning of the separation of the two chromatids, which form one chromosome in the middle. And then move on to the third process, which is Anaphase. Move on to Anaphase. Anaphase 2 is where the chromosomes separate, and then we label them as two individual chromosomes. As the chromatids separate, now we label them as two chromosomes. It's no longer one, it's like this one, where it was together. Where it was together, like that one. Yeah, label it as two. So basically in these two cells, there's four in total, four chromosomes. And then we move on to the last one, the telophase. The last one is telophase, where the final product is 4 haploid cells. So you get 4 haploid cells from the whole process of meiosis 2. Initially from meiosis 1, you got the 2 cells, which you started this process with, and then you end up with 4 in meiosis 2. Yeah, that's all.
Thank you very much for that contribution. All right, can we have men from Skyline to come and give us a contribution? A round of applause, please.
Okay, good morning once again. I think my contribution will be short this time. I think when it has to do with the topic of meiosis, as learners, you have to understand the different phases of meiosis and in the form of diagrams, how they are presented to you. Because if you are presented with a cell at a particular phase of meiosis, and in your mind you can't work it out which phase of meiosis are you being presented with, you are going to struggle with answering the questions. So if you remember, whether it's prophase 1 or prophase 2, and the nuclear membrane is drawn as that dotted line, and the nucleolus is also drawn as the dotted line, then you know you can be able to deduct whether the cell is in prophase 1 or prophase 2. Remember the key event, crossing over, which contributes to genetic variation. You should know by now when the chromosomes are being shown to be in process of crossing over. Then you would know that that particular cell is in prophase 1. And then if the nuclear membrane or the nucleolus, they are still drawn with that dotted line, but the chromosomes or the chromatids rather appear to be in different patches already, then it would be easier for you to reach a conclusion that the cell is on prophase 2. Now, the following phase would be metaphase. And metaphase 1, you know that it is the whole chromosomes lining up at the equator, and they line up in pairs. So it would be easy for you to conclude in your mind that this cell you are presented with is on metaphase 1. And then for metaphase 2, it is still whole chromosomes lining up at the equator, but they are not in pairs. Now, anaphase 1, it is whole chromosomes being pulled to the opposite poles. And then anaphase 2, remember, just before anaphase 2, it is the centromere that splits, and as a result, it would be the chromatids now that are pulled to the opposite poles. And only in anaphase do we refer to them as daughter chromosomes. And then now we have the last phase, which is telophase 1, where you are going to have only 2 daughter cells. And then telophase 2, where you are going to have 4 daughter cells in total. And then you have to know the key processes that contribute to genetic variation, which is the first one, crossing over, that takes place in prophase 1. Crossing over will not take place again in prophase 2. And then you have random arrangement of chromosomes that takes place in metaphase 1 as well as metaphase 2. And then you also have random assortment.
Eish.